Our third group, and the last one that we'll have before today's break, is the group that is mentored by Drs. John Ma and Todd Hammond. So Yu Ren and Bo Forster will be presenting their work on determining gravitropic sensitivity, a mathematical approach.
As he said, I'm Bo Forster, this is Yu Ren. We are studying gravitropism in plant roots. To start out, we'd like to thank our mentors, Dr. John Ma and Dr. Todd Hammond, and also Truman MathBio program at Truman State University in the National Science Foundation. So to give a brief overview of our presentation, we'll start with some background information on what gravitropism is. Move on to the starch-statolith theory, which is a theory that attempts to determine the mechanism of gravitropism, and then move on to auxin, which is the chemical involved in plant growth and its role in gravitropism. And then we'll discuss how to quantify gravitropism, and move on to the progress to date of our project, and then finally discuss our objectives for the summer and the methodology of our mechanism.
Okay, so first of all, gravitropism is the orientation of plant growth indicated by the gravity vector. As you can see, this arrow shows gravity, and as you all may have seen, usual plant growth is determined by roots that go into the ground while shoots that go up into the air. And roots that go downward and with gravity is an example of positive gravitropism. And then shoots that go upward is an example of negative gravitropism. As you can see in this flower pot, when it's tilted on its side, the shoots still go upward. And when it's tilted upside down, the shoots still have the same growth pattern. And as you can see with this root, it goes downward when it's tilted to the side. And gravitropism is essential for the normal growth of plants, as due to the behavior patterns of roots going underground, be able to uptake water and nutrients, while the shoots can go up into the air to perform photosynthesis and uptake CO2. So the major theory about the cause of gravitropism is the starch-statolith hypothesis, which postulates that the gravity perception in plants is mediated by the sedimentation and pressure exerted by starch-filled plastids within the gravity-receiving columella cells of the cap and roots. And these columella cells are in the central region of the root caps. And as you can see, these are the little statolith, which statolith are specialized aminoplasts in the root caps that are the special starch units. So these are denser than the cytosol around it, so these are affected by gravity and sort of sediment in whatever orientation that it's put in. And the statolith then interacts with the plant cells' endoplasmic reticulums and triggers a signal transduction way, promoting organ curvature, and it redistributes the chemical called auxin. The statolith in the root columella cells settle to the bottom towards whatever way you put it, and this is one example is it's an Arabidopsis root cap, and as you can see, the root cap's gravity sensing region is different than the actual elongation zone, and this curvature in the elongation zone is measurable, and that's what's significant to our project. And in this picture is actually an algae cell, which the algae root cap, which is different than usual roots in that it's just mainly one cell that houses these units. And these aren't actual statolith, but these are high-density units similar to statolith in higher type of plants. And these have the same growth patterns as they sediment. When you put it onto the side, it sediments towards the bottom and causes the curvature in the roots. And this picture, it shows this one is a vertical orientation of a root, and as you can see, the aminoplasts or the statolith settle towards the bottom, and there's normal uniform distribution on the the endoplasmic reticulum, and it grows normally downwards. But when you flip it onto its horizontal orientation, it causes an unequal pressure onto the ER, and it causes a redistribution of the auxin that runs towards the bottom. And this redistribution of auxin has been found to be greatly significant in causing gravitropism and curvature in plants. And one way to explain this is the colony wind theory, which proposes that gravity stimulation promotes the lateral transport of auxin across the gravity stimulated organs, which it causes the auxin to accumulate on the bottom of these organs. And this is one example of the effects of auxin and the colony wind theory. This is actually a shoot cap, which has been cut off. and 2 outer pieces has been put onto the shoe cap to absorb the chemicals inside of it, which in the target chemical in this case is oxygen. The one at the top, according to the theory, there should be more oxygen at the bottom of the cap than the top, since it is tilted on the side. So the side with more oxygen is transferred to the shoe, which has been depleted of its cap. And then, as you can see, in comparison to the piece with less oxygen, there's more curvature, and it supports this theory in showing that there's more oxygen on the bottom part of the cap than on the top. And actually, just a side fact, if you cut off the root cap or the shoot cap of a plant, the part will still grow normally, except there won't be any sensitivity in curvature. So that also is a support of the effects of oxygen on gravitropism. So what oxygen actually is a simple chemical that promotes the elongation of stems and roots, and stems, shoots, and roots, and also it promotes other developmental processes in different plants. Almost all plants make this chemical naturally, and the 3 different types of natural oxidants include IAA, 4-CLIAA, and IBA. And the one that we're concerned with mainly is IAA because it's the most prominent one and it's the most physically active. And this is the organic chemistry structure of the IAA that we will be working with in the labs. The main mechanism to describe oxygen transport in roots is the fountain model, which in vertically grown roots, the oxygen is transferred through the vasculature, and as you can see, these black arrows show the movement of oxygen through the root cap, and then the gravity vector is determined by this blue arrow. And then it transfers through the vasculature into the root, where it accumulates in the first layer of the columella. There it is transported laterally to more peripheral tissues, then to the site of response, where it inhibits cell elongation. But once it's gravity stimulated to its side, the lateral redistribution of oxygen occurs preferentially to the bottom of the cells. And it results in a lateral oxygen gradient, which is transferred to the site of response, where it promotes a downward tip curvature. In normal concentration of oxygen is actually very low. In a very low concentration, it promotes growth in the plants. But then actually, once it hits a certain gradient, once it hits a certain concentration high enough, it starts to prohibit growth. So that is actually what causes the curvature in the plants. So once high concentration of oxygen is collected on the bottom, this bottom side of the roots starts to grow a lot slower than the top part. So then the top part starts to elongate more than the bottom part, and it causes the downward curvature as you've all seen. We can measure gravitropism in 1 of 2 ways. The first way is measuring the response to gravity by roots of different ages. This will be done by observing the angles of curvature at different time-lapse intervals and comparing the rates of curvature. As you can see, this is from last year's team's results, and they've done, in this example, there's 2 different time periods, the 22-hour roots and 30-hour roots. And then the second way is by measuring gravitropic sensitivity of roots of different ages. This will be done by determining the presentation time of the different roots. And the presentation time is defined as the minimum duration of exposure to stimulation in a 1G environment for the induction of gravity response, which sounds kind of confusing, but it's basically the amount of time that you need to stimulate a certain root for in order for it to respond to gravity, meaning that the minimum amount of time that you need to stimulate it in order for it to start showing any signs of curvature. So then once you get these different time intervals, you can calculate the presentation time by plotting the root curvature, which is here, against the logarithmic number of the gravity stimulation time, which is the induction time, and it's the amount of time that we allow the roots to be affected by a 1G environment. Then you extrapolate the regression line to the x-axis, which is also the region of zero curvature. And this point where it hits the x-axis is the estimated duration of time that you need to gravity stimulate the roots at a certain age in order for it to show any kind of gravitropic response.
So both of the ways of measuring gravitropism depend on quantifying the curvature angle in the root. And normally this is done manually. There are several drawbacks to this. One, that it's slow, and 2, that it's subjective, and therefore the measurements are less accurate. And so developing a mathematical model for computing the curvature We first identify the roots in a picture using an image segmentation technique called seeded region growing. And then the perimeter and medial axis of the roots are identified. Once we've identified the perimeter of the root, our software steps through each point on the perimeter and looks across the root for another point on the perimeter and finds the closest one and plots the midpoint between those two points. And so essentially, what it's doing at each point is finding a maximal circle and plotting the center of the circle. And then the centers of the circles are smoothed out, and that results in the medial axis. And then mathematical curvature is computed at each point along the medial axis, and then the overall root curve angle can be interpreted from that information. And also we compute the cross-sectional area using green. So the progress of the project to date, so far we have developed an algorithm for quantifying root curvature, and also we have found that as roots age, both the sensitivity and the response to gravity decreases. And so our objectives for this summer are to, one, refine our algorithm, and possibly to do this we would draw on alternative methods of quantifying root curvature. And then also we would like to organize our data into an accessible format, and this would help us with our data analysis and our eventual results part of our research.
Okay. On the biology side of the objectives, first of all, we want to determine how root growth and response to auxin as root ages. As I said before, auxin is a significant part of gravitropism, and we're going to try to figure out exactly what effect it has on it. Because we found out from last year that roots do become less sensitive as they age. We also want to determine how auxin affects gravitropic sensitivity and response as root ages. So one of our main focuses this summer will be using the chemical auxin in determining these causes. So the methodology we'll be using will still continue to use the flax seeds as the previous team did, due to its quick germination time. At a full 24 hours, you can tell a complete growth in the roots, and significant enough for us to observe the curvature. The seeds will be germinated in petri dishes filled with agar at the growth medium. And unlike last year when they used the germination paper to put the seeds on, we with the physics department created a mold in which will be placed directly on top of the agar plates when we make the agar plates. And these will make 12 lines of little wells that fit the seeds, the flax seeds directly, which after measuring the dimensions of the flax seeds, we found them to be 0.2 centimeters by 0.5 centimeters. So after doing that, we will germinate the seeds directly inside of the agars. And this has a couple advantages. One main one is because we have to run these pictures through the program, we want to eliminate as much background noise as possible. So the germination paper before, it has a different color than the agar background. So in this case, there will be less distortion and less noise in the background, so the program can run more efficiently. Since each mold has 12 different wells, and we can fit a maximum 3 molds on the petri dish, we can have a maximum 36 wells per petri dish, meaning we can germinate 36 seeds at once. So the first part of our experiment, we will be trying to determine the growth response of roots to auxin at different ages. The first thing we'll do is germinate the seeds on the agar plates for different time durations. Then we will treat the roots with different ages with the auxins at different concentrations. And after treating them, we will determine the root elongations for each age and the concentration of IAA. We'll set up a database as Bo has said before, and we will make graphs of all the different calculations. And then we can compare the roots of different ages. These are just 2 samples of. Actually, this is one root that's been kept. Both of these have been germinated for 24 hours. And this one, after coming out, has been treated with a concentration of auxin that would be inhibitory to growth. And this one has been treated with a control substance that usually that would not have any effect on growth rate. So as you can see, it's not as easy to tell without using a program to calculate the exact growth rate, but the one on the right grew faster than the one on the left. And this growth rate, this different growth rate is what we use to calculate the response of roots to the auxin. and as well as the age. For the second part of the experiment, we will be determining the auxin effect on the gravitropic sensitivity and response of different root ages. The first thing, same thing we'll do before, germinate the seeds, and then we will treat the roots. We will treat the roots with the auxin before taking them out of the incubation chamber, and then we will flip it onto the side at a 90-degree angle for a different amount of gravity-stimulation times. The ones that we'll be using is 5, 10, 15, 20 minutes, because those are the ones that we've tested in the past to be most significant. After treating them with gravity stimulation time, we will put them on the clinostat, which in this case, it will stimulate a zero gravity environment. And as you can see, in the normal settings, when you flip a plant on the side, the shoot will tend to go up and the roots will tend to grow downwards. But once you put them on a clinostat, everything grows uniformly because going back to the starch statolith hypothesis, the aminoplasm and statolith never have time to completely sediment on one side because it's constantly rotating. So then it imitates a zero-G environment, and the plants never really get a time to settle, and then thus it just keeps going forward straightwards. So in our case, the only gravity stimulation that it has is before it goes onto the clinostat when it's rotated at 90 degrees. And then we will keep it on the clinostat for 3 hours because that is the time that we found to allow the roots to have maximum potential for curvature. And then afterwards, we will use the we'll take a picture of the roots and use the computer program to determine the actual curvature in these plants. And then, as I said before, we will plot these points on a graph, and then we will extrapolate the points back to the x-axis, and this will show us the presentation time and what's the minimum amount of time that we need to introduce the roots at each individual age in order for them to show a gravitropic response. So, with that,
we'd just like to thank again our mentors, Dr. John Long, Dr. Todd Hammond, and to acknowledge the Truman State University and the Truman MathBio program. And again, we'd like to give a big thank you to the National Science Foundation for funding our research. Thank you.
Questions for this group?
At one point, you had talked about a response in an algal cell. In algae?
Oh, in algae, yes.
And I'm wondering, first of all, if you know anything about what kind of alga it is, like was it a single-celled alga or not?
It was a single-celled green algae, yes.
Okay, so what's the point biologically? In other words, do you know anything about the relationship between this response in a single-celled alga and anything that has to do with how the algae makes its living?
Basically, with showing this, algae has, the curve rate has a significant similarity to the plants that we will be using. Not in everything, because it doesn't have exactly statolith, because it is in its simple structure. But these little particles are actually mineral-rich particles that mimic the functions of statolith and has the same sedimentation patterns as the plants that we will be using. So I'm not quite sure about the exact lifestyle and life cycle of algae, but in this case, the rates of curvature, the patterns of curvature is the same as the ones we will be studying.
You know, partly what I'm getting at is there's a good adaptive story that you can tell about why having a gravitropic response might make sense for vascular plants. And I'm wondering whether a similar sort of story exists for algae. I mean, there isn't any apparent reason that I could think of why it would make any sense for an algal cell to have that response.
That's true.
And you know, kind of what is it doing for it in this environment? That's really what I was driving at.
I understand.
It's worth thinking about. You might want to go back and check out the paper and kind of see, because just because the mechanism exists doesn't mean that it is doing anything that is like what it's accomplishing in vascular plants.
I understand. It doesn't really have the same.
Yeah, I mean, it might be closely related, and then it might be completely not related.
For your curvature versus time graph, do you make a graph for each of the different roots, or are you taking an average?
For all the different roots, we won't be making one for every single root, because some roots actually come out with more distortion than others, as in they don't curve as uniformly as other ones. But we will be that would be actually to try to make a database of as many uniformly growing roots as possible on each sample. That's why we're doing so many samples on each petri dish, as to get the best response from the roots as possible. So each individual point is actually a different root that we're looking at, so they're not really an average. So actually for each petri dish, potentially we can have 36 different samples from each growth.
Right now, it doesn't actually use the seeded region growing. It goes through and does a statistical analysis of the pixel color, and that's it could be better right now because it picks up a lot of noise, a lot of the glare pixels, and other pixels that are close enough in color to the root tissue that it identifies them as root. We haven't yet completed the seeded region growing, but we're hoping they'll do a lot better job of identifying the roots. Yes.
You say you're calculating cross-sectional area here too?
Yes.
Are you using the same method?
It actually uses application of Green's theorem on the perimeter of the root that we identified.
Oh.
So one of your experimental treatments in the experiments is exposing the seeds to different concentrations of auxin. So you're putting that in the agar, presumably?
No, actually, we will be germinating the seeds with the agar plates in a germination chamber. It's set at 23 degrees Celsius, and then for a set amount of times, 22, 26, and 30 hours. And then after the germination time, we'll be submerging them in the auxin solution while they're inside of the petri dishes and directly inside the incubation chamber. So then after we take it out, we'll rotate it on its side and let it be exposed to gravity stimulation. But it will be done still inside the chambers.
So the seeds are getting exposed to the solution, and then presumably they're taking it up.
Yes.
So the treatment is about providing the germinating embryos with
Additional
exogenous auxin
Yes.
beyond that which they would typically produce.
Yes.
So what do you know about whether or not A, they take it up, and B, it actually acts like endogenous auxin? You know what I'm saying?
Yes.
Yeah. Okay.
First of all, we've done a couple trials, and we've actually determined that there's a significant difference in the growth rates between the roots that actually uptake auxin and that we've submerged, and the roots that don't uptake auxin and that just grow under normal. Okay, let's see. I have a slide that shows. There we go. In roots, we know that in low concentrations of auxin, 10 to the negative 10 to 10 to the negative 9 molars, it promotes growth. But the concentration greater than 10 to the negative 6 molars inhibits growth. And this has been a well-researched area. So people, there's wide support for this. And then what we're testing is beyond the 10 to the negative 6 molar, at what rate does it actually inhibit growth? And what's the comparison between that and natural auxin concentrations?
So do people know anything about, does it just kind of distribute itself throughout all the cells uniformly, or does it tend to accumulate in particular cells because those cells are somehow especially sensitive to taking it up, or certain cells tend to export it and other cells tend to sequester it? Do you know what I'm saying?
Yes. Yeah. It's mainly produced in the root cap region, and as I said before, it's transported throughout the cells by the fountain model, in which usually when it's tilted downwards, it just secretes uniformly. But then once the the statelets come in contact with the endoplasmic reticulum, it creates the transduction pathway, and then that also affects the auxin and allows it to sediment sort of in the same way towards the bottom of the root, and then thus it inhibits the growth towards the bottom region. So in that way, it mostly is focused around the root region, around the around the response, side of response in the root caps. Does that answer your question?
Yeah, kind of. I mean, you said what you said now, what you just said before, and I understood that part. I guess what I'm getting at is this. It's a single molecule. It's quite clear from your description that in order for that to work, cells have to be differentially sensitive both to its export as well as its sequestering. Otherwise, the stuff would just move down its concentration gradient just like any other diffusing material would, right? So if you're thinking about it being taken up by an embryo, you would have to know, it seems to me, a priori, or test where the stuff is going and where it's ending up, because otherwise it's just a simple diffusion model and it ought to be everywhere, and there's no reason to believe that it's going to have the kinds of effects that you might be claiming it would have. And I have little doubt that this has been really looked at pretty carefully in the literature. I just don't know because I don't read this part of the literature, so I was just exploring with you what you might know about it.
I don't know about time getting to this, but this is one of the slides we kind of took down. This is actually how auxin travels across the membranes and walls between cells, and it's mostly focused in the meristems of the cells. And basically, mainly that's what we know about it as of now. It's mostly focused around the root cap region, and it is concentrated to certain cells, the columella cells, and it doesn't really go into the other vascular tissues outside of that. And then because of that, it allows it to perform traveling across the membrane between those certain specified cells.
Would there be any interesting results from studying plants to extra-gravitational force and observing the effects?
Well, our project actually is right now just on a small scale of possible future studies, because there's a bunch of different tropism studies that has to do with the growth rate of plants. There's phototropism, there's hydrotropism, and gravitropism is one of the significant ones. And it's just to allow us to better understand the growth rate of these different plants for one possible future study is the growth of plants in space. Because for now, if you bring a seed into outer space and allow it to grow there, it will not grow because they're under zero-G environments. The roots, if they don't have any gravity stimulation time prior to coming out of the shells, then they're not going to grow correctly. They're basically not going to germinate at all. So in our case, we're trying to determine the minimum amount of time that you need to actually stimulate the roots, stimulate the seeds under a normal gravity environment before you can introduce it to a zero-G environment such as space for it to actually grow optimally.
Let's thank this group again. We'll now have a 15-minute break. There are refreshments. Feel free to use the restroom, and we'll reconvene at 10 o'clock.